Good evening. Can you hear me? Good evening, everyone. उसने बोला ये बंद करने का। ये कहाँ से ऑन होता है? Good evening. Am I audible? Good evening, ladies and gentlemen. On behalf of the Director, staff and scholars of the National Institute of Plant Genome Research, New Delhi, I welcome you all here. Today we have Professor Sanders, can you hear us? Professor Sanders, can you hear us? Professor
I can hear you fine. Can you hear me?
Yes, yes. So we are about to begin.
Sure.
Good evening, everyone.
स्टार्ट वीडियो कर लीजिए.
Start video.
ये नहीं टच नहीं होगा. ओके.
Good evening, ladies and gentlemen. On behalf of the Director, staff and scholars of the National Institute of Plant Genome Research, New Delhi, I welcome you all here. Today, we have gathered here to celebrate the 24th Foundation Day of our institute. 23 years ago, on 30th November 1998, DBT established NIPGR as an autonomous organization with the mission to undertake research of high caliber in basic and applied plant biology. Since then, the institute has grown and is now well known for its contributions in cutting-edge areas of plant research. This day also coincides with the birth anniversary of the eminent scientist Sir J.C. Bose, whose scientific intellect and patriotism are a source of inspiration to all of us even today. Hence, on this occasion every year, the Institute takes great pride and pleasure in organizing a Foundation Day lecture in the memory of Sir J.C. Bose. So once again I welcome all the distinguished guests from various organizations who have joined us in person and virtually to celebrate this special day. A very special welcome to Professor Dale Sanders from the UK. I also welcome all the members of the NIPGR family who have gathered here to grace this occasion. I now invite Dr. Shubhra Chakravarti, Director NIPGR, to kindly come and grace the dais. I also request Dr. Alok Sinha, Scientist NIPGR, to come to the dais. As is customary, we would like to begin the event today by lighting the auspicious lamp. I request Dr. Shubhra Chakravarti and Alok to please join for the lighting of the lamp. To begin today's proceedings, I request Dr. Shubhra Chakravarti, Director, NIPGR, to deliver her opening remarks.
So, Honorable Dr. Rajesh Gokhale, Secretary, Department of Biotechnology and Chairperson of Governing Body NIPGR, all DBT Autonomous Institute Directors, Professor Dale Sander, Honored Speaker of the Day, dignitaries present, colleagues from NIPGR, DBT and other institutes, the young investigators, dear students and researchers, staffs of NIPGR and the participants who joined either offline or online, a very, very good afternoon to you all. On behalf of the NIPGR family and my own behalf, I extend a very hearty welcome to all of you on the 24th Foundation Day ceremony of our institute. Dr. Sabat Bhatia already mentioned about the genesis of the institute. Just to brief, since we have amongst us today Professor Dale Sander and many other colleagues from abroad, and for the young students who have joined this year as our first new PhD batch, just to give you a brief overview on the genesis again. The seed of the institute was sown actually long back, almost 24 years back, on the day of 30th November 1997, and that was the year of 58th, the anniversary of India's independence. Today we are celebrating started celebrating the 75th year of independence of our country, while NIPGR is possibly next year embark on its 25th year of existence. So, and also this was on the very day of the Sir J.C. Bose's birthday, and who doesn't need any introduction, at least I know in this audience, because we all know his contribution, although he was a physicist, but his contribution in the area of plant science and particularly to the plant's perception. So in that case, the institute was set up with a mandate to educate, to promote the science and particularly the plant science to the young students and the researchers, and coordinate research work amongst not only to the Indian institutions or universities, but also we have a lot of collaboration that we have grown for last 24 years with international partnership as well. And this is for Dale, that I have already talked to you, that that NIPGR has now 2 years back has signed an MOU with also the UK participation. I'm looking forward that when that actually come into existence, due to the pandemic, possibly the program got little delayed, but we are all eagerly waiting to start that program with all the cooperation of the UK partners and also other DBT autonomous institutions in the field. So just to tell you that 24 years back when the institute started, it started only with 16 employees, 7 faculty members and 8 technical staff and 1 administrative staff. Today, after 24 years of our existence, I'm very happy to tell you that we have grown 30 times, and now our whole strength is around 485 of us, where we have 30 faculty, we have more than 135 PhD students currently enrolled in the institute, and also we have more than around 250 researchers, including postdoctoral fellows, with 29 technical staff and 20 administrative staff and 16 multitasking staffs. We are located in a prime location of the capital of India, that is New Delhi. Why I say it is a prime location? Because we are situated in a South Delhi research cluster. We are actually in the lap of Jawaharlal Nehru University, and with which the academic program of the institute is affiliated from its start until we continue to do the same. Not only the JNU, but also in the same campus, we have our another sister autonomous institute of DBT, that is National Institute of Immunology. We also have opposite to us Delhi Indian Institute of Technology, that is IIT Delhi, and also some more institute nearby. Therefore, the institute is very much well placed in an academic atmosphere where students and researchers and faculty can interact with each other across different institutions. And at the same time, our whole research in last 24 years have been actually driven by groundbreaking approaches to the research. Not only that, with the cutting edge technology platforms, most importantly, with the dedicated scientists and students and staff that we have in the institute that help the institute to be established as the premier plant institute in India, not only in India in the cutting edge of the plant molecular biology and biotechnology, but also starting from the inception, the NIPGR research is actually towards the basic research, and any lead that comes very keenly and very passionately, the institute is trying to translate it for the benefit of the society. And NIPGR presently carry out in 6 defined research area broadly, that is computational biology, genome analysis and molecular mapping, plant development and architecture, molecular mechanism of abiotic stress response, plant immunity, plant pathogen interaction, molecular breeding, nutritional genomics, and to develop crop variety starting from molecular breeding up to the gene editing or transgenics and emerging as an area of Plant Genomics Institute. And although it started with structural genomics, but now institute is actually geared up to deliver and ask research questions in the area of functional genomics using more of the other omic technologies, and also many interaction map and kind of a related network hypothesis driven research. I don't need to emphasize more that plants are fantastic organisms. They give us oxygen, they give us food, and they actually give us all the energy that we all live on this very wonderful planet Earth. And therefore we study the fundamental processes in the plant in the 6 domains that I have shared with you, and that how plant grows, how plants survive under the different stress condition in the field, also how all these macromolecules like protein, gene and metabolite can interact with each other, and with a system biology approach. And also institute just got one very young Ramlingaswamy fellow who has first time developed the structural biology lab last 2 years back in the institute, and we are looking forward lot many from him, and it is a very nice addition with Dr. Vinit Gaur with us today, that we would be able to already embarked onto the field of system biology, including structural biology, which is actually a very important area in today's work. Also, we have started thinking of making chemical biology. Dr. Jyoti Lakshmi came here from Germany, and she has started it. She has set up a very nice metabolomics facility in the institute, and we are also trying to expand in the chemical biology area in addition to the system biology approach or the structural biology that I mentioned right now. And with the primary goal to gain knowledge, many of our research findings are translated, as I already mentioned, and that constant learning and improving with commitment and dedication and fresh energy has always driven the research of the institute, and we look forward for some same vigor from our faculty, our staff and our student, in cooperation with all our colleagues from national labs as well as international partners, to take the institute in much better height. And therefore, today NIPGR's vision is to be recognized as a center of excellence, not only in the Asia Pacific region, but in the globe, and also among one of the top plant science institute in the world. And to fulfill the vision, today our mission is to continue to call for research and innovation to have a transformative impact on the society. Also in addition to the research and many other activities, NIPGR also has a very important activity, that is our outreach program, where more than 1,000 students come and visit the institute. They go to the different facility, they interact with the faculty and staff and students. However, for last 1 year, 2 years almost now, that we could not make any physical presence of them. But therefore, last year onward, we have started a kind of a program which we call now NIPGR webinar series, and there are in every series one of the scientists from the institute, along with one national expert in the field and one international expert in the field, gives a talk in different areas, and those are all open to everybody across the globe, and it all are online. So along with that, DBT has started one program which is known as Science Setu, and in that case it is to encourage school level students and college level students to encourage to come in the STEM field. And therefore, using this kind of outreach programs, NIPGR is trying to reach out from the school to the college, to the university and the research institute to encourage more and more students come to the STEM field, particularly, as you understand from our institute name, in the plant area. So because we see decline, and this decline is not only the problem of India, but I'm sure it is a problem of the globe, and we need to encourage people for the STEM, and that is where this program is actually destined to come out with better things. We hope for invigorating the interest within our younger generation to the plants, which are, I said, a fantastic organism. Now, this year, during this difficult time, also NIPGR would join. India started the India International Science Festival for last 7 years, and this year it's going to happen in a kind of a hybrid mode in Goa from 10th to 14th December this coming month. And in that case, our outreach program also will put up a stall, and all the students and everybody can also join online to see what all infrastructure facility, what all lab, what all work students and faculties are doing in the institute. They can have a virtual tour, and also there will be many models and many posters that would be put up in the stall to give a glimpse about the institute research. And then also there will be stalls of NIPGR. People who would be visiting IISF can also physically visit there. Those who are joining online, I welcome all of you, and also to tell your friends and colleagues to go and go to the NIPGR stall to understand more about NIPGR and its research. I would like to take this opportunity to mention our deep sense of appreciation to Professor Sander. We were expecting him last year, however, due to certain reason, Dale could not join us, but we are all so happy and glad, and it's really our honor, Dale, that you are amongst us as our, most valued speaker of the day. And also, I take this opportunity to actually tell our deep sense of appreciation to the audience who have joined either physically or online and participation with us in this very important occasion. So I would like to say institute started, and that time it was a bud. Today it's full bloomed institute, and we expect many more from our students. We are trying to give all the platforms, all the facility that they need to do a world-class research. I hope the students and faculty and staffs are all happy with the institute, and we institute look forward from them much more in next few years and in future. I would like to close by congratulating the entire NIPGR family for their hard work, dedication and sincerity, and to keep the campus a very, very vibrant campus within the chirping of the birds all throughout, and having such a great campus within the, the polluted city of Delhi, having such a green campus. It's really a pleasure and privilege to be there, and more so during this difficult time. Wish everyone a very happy Christmas and a new year, which is a safe new year, I hope so, after Omicron has come, and till we meet the next time. With that, I would close here, and Dale, thank you very much. And I hand over the mic to Dr. Bhatia.
Thank you, madam, for your kind words of encouragement. Your scientific perspective and long-term vision will steer the Institute to great heights in the near future. On this special day, I have the honor of welcoming a very distinguished and eminent scientist, Professor Dale Sanders, Fellow of the Royal Society and Director, John Innes Centre, UK. Professor Dale Sanders is a project leader and director of the John Innes Centre in Norwich. His research explores the mechanisms of heavy metal and calcium transport in plants and the physiological signals that trigger these processes. Dr. Sanders received his BA from the University of York and his PhD in 1979 from the University of Cambridge in plant physiology. After postdoctoral work at the Yale University School of Medicine, using fungi as model systems for understanding how plants absorb mineral ions, he joined the University of York as a lecturer, where he went on to become head of Department of Biology before joining the John Innes Centre. He is a Fellow of the Royal Society and has received the European Science Prize of the Korbner Foundation. Under his directorship, the John Innes Centre established the Centre of Excellence for Plant and Microbial Sciences in partnership with the Chinese Academy of Sciences. I could go on about his achievements, but would not like to come in between you and his talk that we have been eagerly waiting to listen to. Today, he will be talking to us about plant nutrition, elements for crop and human health. Professor Sanders, over to you.
Thank you so much. Can everybody hear me? Can you hear me? Can you?
Yes, Dale, we can hear you.
Yes, okay. Okay, great. I'd just like to say a few words in response to what I've heard. I am so honoured to be here today, and a warm welcome to everybody and a good afternoon to you all. It's a really an auspicious occasion, and I hope that I can stimulate you with some science, but also I'd like to say a few words near the beginning of my talk on UK-India connections. So this first slide illustrates, I think at the top, if you can see my cursor, the range of topics that I would like to discuss today. So that goes from the molecular to the ecological, and from plant nutrition to human nutrition. I'd just like to say a few words about Professor Bose, and this has already been commented on. Of course, he won a Nobel Prize in physics, and what more could one want? Here's a lovely cartoon of him from 2016, celebrating his 158th birthday. But what I also wanted to mention was his interest in plant biophysics and plant electrophysiology. And that was something that really inspired me when I set out on my research career. This was only a sideline of Professor Bose's, but it did illustrate the breadth of thinking that somebody as brilliant as he was had in doing interdisciplinary science and thinking beyond the realms of the physical sciences and into the life sciences. And what I wanted to was just to say how this influence happened for me, and I'm going back many years now to think about my inspiration of biophysics as a life scientist to the work that I did. And in this paper that now goes back 40 years or so, we were the first to show that the uptake of ions by plants is accompanied by and powered by a proton gradient. And we accomplished this through electrophysiological approaches, and this provided something of a paradigm for thinking about how plant membranes energize themselves. And in a subsequent paper a couple of years later, when I was at Yale, we investigated the properties of a model fungus, Neurospora, and examined the electrophysiological characteristics of the plasma membrane, and were able to show through this current voltage analysis that actually when you acidify the cytoplasm, the proton pump is activated. And I think none of these kind of approaches would have been achieved without the fundamental biophysical thinking that underpins our current understanding of biology. So I just wanted to say a few words before I start on my science about our interactions with India. So I'm from the John Innes Centre, as you know. Janaki Amal, I think, was awarded the first PhD in botany from India, and she worked at the John Innes in the 1930s and 1940s, and we have indeed established a studentship from developing countries in her honour, and to honour our inclusivity and diversity thinking. And we've pulled that forward through thinking with DBT to bring in students and postdocs. And here's an example from this year, Dr. Nitin Kambal from NIPGR, who joined us to work with David Sung, studying starch metabolism and enhancing the starch value of cereals. I've visited India many times, and it is my most recent visit, and I'm so sorry not to be there with you in person today. Here we are in the Punjab, talking with Professor Harminder Singh Sidhu about the issues of heat and drought with respect to wheat and crop production more generally in India. And that was really such a productive visit in 2016, and established really strong relationships, I think, in plant science between my own institute and the UK more generally, and with India and with DBT. And as part of that visit, we hosted, made plans to host a visit for females in agriculture and agricultural research to the John Innes Centre, to Cambridge University, and to the Rothamsted Research. And that was a really happy occasion where lots of social interactions were established, as well as lots of good research interactions. And I think that was a measure of our commitment, really, to establishing strong links with India. I just wanted to say a few words now about the John Innes Centre itself. So we work around 4 fundamental research programs. One of those is on plant health, so that this image here shows spores coming out from a wheat leaf of yellow rust, false coloured of course. So we have a big interest in plant pathogen interactions, and I'll come on later in my talk to discuss a little bit about my own work with insects there. As I've mentioned in the top right-hand corner here, we work extensively on wheat and have tremendous international interactions there. In the lower left, this photo of Arabidopsis indicates our commitment to understanding plant developmental genetics. And then in the upper left, this image represents our interest in plant biomolecules and using plants as systems for expressing enzymes and pharmaceutically active molecules for the benefit of human health. So we have these 4 programs, and they form the centre of what we do. But of course, each group leader within our institute is free to do their own research as well along those lines. Okay, so I'd just like to move on then to talk about elements. And I'm sure you're all familiar with this periodic table that many of you will have focused on in your chemistry years. And I'd like to make the point through this slide that through these elements that I've highlighted, these are the elements that we tend to focus on as biologists. They're the components of proteins, they're the components of DNA and so on. But this is just to remind you that there are many other elements that perform critical functions in plants and indeed in humans. And sometimes these might get forgotten among the bigger picture of proteins and nucleic acids. And I wanted to talk today in terms of my research in where we think we can go in understanding how these elements can be used to improve human health, and ultimately how we can understand that elemental production or the elemental use of elements to understand where plants grow. And I'll finish off my talk with that. And of course, just to mention, I've highlighted in red here elements which have no known biological use in plants. Of course, sodium is a major element for animals, but arsenic, cadmium and lead, of course, toxic. And I'll address issues of elemental toxicity in plants and how we can think about using plants to remediate that. So just to go to a broader picture now, where we're coming from, where I'm coming from in the John Innes Centre is how do we feed the world sustainably in the next 30 years? We're expecting another 2 billion people on the planet. How is that going to be achieved sustainably without ruining the environment? And this just shows you projected numbers over the coming years in terms of global population. The challenges are multiple. We have this increased demand for food. We have, as I guess everybody knows, an increased demand for water. And we also need to think more about the net zero and energy usage in the way that crops are produced. And of course, agriculture agriculture is a major CO2 emitter. Some estimates give that number as much as 30% of CO2 emissions coming from agriculture. So how do we get to think about that in a sustainable way? I wanted to start, before addressing those bigger questions, to give you a little bit of a story about how I got thinking into this area. And it wasn't particularly in any of these bigger issues, but it was more from the point of environmental pollution. And I just wanted to share with you, I've lost my slide here. The notion which we have, I hope everybody can see that, in Europe particularly, and the USA, of terrible pollution that has come through engineering and metal production commercially. So on the left-hand side is an image of an old zinc smelting factory, and on the right-hand side are the consequences of that, which lead to desolate land, which is incapable of growing plants, and which cannot support life. So a number of years ago, I started looking at the properties of membrane transporters, which are likely to transport metals such as zinc. And I came across this family of cation diffusion facilitators, which are ubiquitous in all plant kingdoms. And there's an image, a generalized image there of what they look like. And they transport, as you can see at the bottom there, they transport a wide range of so-called heavy metals. And we conducted a phylogenetic analysis of these transporters some while back, and were able to demonstrate that actually you can categorize these transporters along the lines of their metal specificity through the phylogenetic analysis. So this zinc clade here is not exclusive to zinc, but zinc is the primary metal that's transported, similarly with the manganese clade and the iron and zinc clade. And one of the things that I reasoned was that we might be able to exploit those transporters to remediate or clean up soils to enable plants to grow on soils which were contaminated with heavy metals. And what I wanted to, before I start on that story, what I just wanted to say was that to provide you with a sort of a plant sciences 101 picture of a plant cell, and to remind you of the existence of the vacuole, and which is a crucial organelle which often gets forgotten in terms of thinking about plant cell biology. So of course, if you look at the image of a plant cell as it's taught to biology undergraduates, they're taught that the plant cells are unique, that they have cell walls, which animal cells don't. Of course, they have chloroplasts. And then there's this rather boring looking hole in the middle of the cell, which is called the vacuole. And what I'd like to do is argue that it has many important aspects in the way we think about plant life generally, and about the more general impacts that we can generate from studying plants. So I'm going to go back now to just discuss how plant vacuoles operate, and you can see here that with respect to the energization of the vacuolar membrane, plants use protons, as I mentioned much earlier, as a energetic force through the hydrolysis of ATP and through the hydrolysis of pyrophosphate to set up a proton gradient that generates this membrane potential and a pH gradient across the membrane, and that can be utilized to energize metal sequestration into the plant vacuole. It can be utilized to accumulate calcium, and I'll come on to that in the last half of my talk. And it can potentially be used to understand how soil adaptation in plants can be achieved in various ecotypes of species. So now to go back to the story about these metal transporters, we showed that this transporter MTP1 in Arabidopsis can locate the vacuole, and that its overexpression confers partial zinc tolerance. So on the left-hand side here, you see a GFP fusion image of MTP1 in a root tip of Arabidopsis, exhibiting localization to the root. And then on the right-hand side, in the presence of high zinc concentrations, tolerance is generated in plants, in Arabidopsis plants, to high concentrations of zinc in comparison with controls which were transfected with the anti-vector. You can see the data there showing that verified by the seedling fresh weight. So this suggested a way then in which one could exploit an understanding of plant transporters to potentially remediate soils and also enable plants to grow on soils which were contaminated with heavy metals. But that understanding led actually to wider and more, I think, deeper thinking about zinc and its role in plant and human health. And this image on the left-hand side shows you that global zinc deficiency in agricultural soils across the world is significant. And if you look at zinc deficiency in terms of people, it is profound in terms of the number of people who are defective in their dietary zinc uptake. And I just wanted to say a few words in the context of human health about potentially the consequences of this. The impact of zinc deficiency on human health is indeed profound. It contributes to over 800,000 deaths a year from infectious diseases, so that those are people who are immunocompromised. Many of those diseases relate to infection through pneumonia and malaria, and particularly in infants. And there are a range of other issues that are associated with zinc deficiency, which was interestingly only identified as an issue for human nutrition about 50 years ago, much after iron was recognized as a potential problem. So why are so many people zinc deficient? And the answer, I would say, is not particularly through the deficiency of zinc in soils in which plants are grown. It's through the deficiency of zinc in the endosperm of cereals, which are quite good accumulators of zinc, but which don't deposit the zinc in the bits of the cereal grain that most people eat. So here's the endosperm of a barley seed, stained with a colour indicator, and you'll see it's virtually deficient, whereas in the Allurone layer, the zinc is plentiful. And here's a similar image for wheat. The embryo here is well stained with zinc. The endosperm is virtually deficient. So we reasoned a while back that we might be able to rectify this deficiency by taking our knowledge of and our thinking about how transporters might remediate heavy metal contaminated soils by expressing them in the endosperm into the vacuole of endosperm cells, and thereby enabling enhanced zinc accumulation in those cells. So this schematic shows the basic thinking there. There's no problem with these cereal plants taking zinc up from the soil. No problem in accumulating it into the Allurone cells, but getting it to the endosperm cells is clearly an issue and hasn't been accomplished. And the thinking behind this then was to express one of these NTP transporters on a vacuole-specific promoter, an endosperm-specific promoter, which would enable accumulation of zinc in those cells. So we were able to demonstrate using this barley HV-MTP1 that it complements as a zinc defective yeast mutant in terms of its ability to grow at high concentrations of zinc, which showed that this was a promising candidate. And we furthermore showed that the zinc transport of the MTP1 localized to the vacuoles of zinc and of plants, and went on to show that you could actually through yeast experiments develop significantly enhanced zinc accumulation in these cells. And this looked really promising in terms of biofortifying crops. So we then overexpressed this MTP1 under a D4-Dein promoter in barley as an easily transformed cereal, and showed, so the D4-Dein promoter is an endosperm specific promoter, showed that there was no visible phenotype in expressing that, and then went on to analyze the grain content of these transformants. And what you can see on the left-hand side here, and again this is with a zinc-specific stain, is that the endosperm is highly enriched in the zinc compared with the controls. So that looked really promising. Sorry, I need to go back. And we were able to confirm this by ICP, OES, which shows that the transformants were significantly enriched. And then furthermore to confirm that with synchrotron imaging of zinc in barley grain sections. And so what you can see in the inset here is the wild type, which classically and I think very beautifully displays this accumulation of zinc in the Allurone layer, nothing in the endosperm layer, and then these three transformant lines showing the zinc somehow being sucked out of the Allurone layer and into the endosperm layer, which potentially enriches this for human nutritional purposes. So to summarize this section of the talk, what I'd like to say is that plants sequester zinc and other metals into vacuoles using a number of transporters. The metal specificity determinants have been identified in my lab among others. Importantly, that zinc is an essential element for life and that zinc malnutrition is widespread in humans, and that overexpression of transport for improving dietary zinc deficiency. And of course, I must emphasize that was very much a GMO approach, and there are thoughts across the globe about how widely we should be applying those technologies. But I think there is enormous potential in thinking about this in terms of analysis of genomes more widely, and exploiting natural or induced variation, and not necessarily using GM techniques. I'm going to skip the next few slides because I'm slightly running out of time. And I wanted to move on to the second part of my talk, which is to think about another element and its importance in plants as well as animals, really. But and that's calcium. So, as many of you, most of you, I guess, will know that calcium is a signaling element which leads to lots of responses in plants in response to antibiotic stresses. And I wanted to talk a little bit about some of the work we've been doing on one defense response, which relates to aphids. And I would say that this is a collaboration that would never have happened had I not been in the institute that I'm in, and it happened over a conversation with my colleague Saskia, who works on aphids. And this is just a summary slide of aphids, aphid biology. They are huge transmitters of plant viruses, a huge threat to plant health. And so we're not just talking about insects here, we're talking about plants and their impact on crops and widespread diseases. And we set up a project to work on this aphid here, which is a very broad feeder, Mysis persicae. It has huge resistance to pesticides. It transmits large numbers of pathogens, and it's very promiscuous in its feeding in terms of plant species that it will dominate. So what was known about the way in which these aphids feed is that they penetrate the epidermis of a plant leaf. The stylet then moves down through the mesophyll, passing effectors which prohibit plant defense responses. And when the stylet finally reaches the phloem cells, the aphid can begin to feed, which it does. And there had long been speculation that calcium might be involved as a signal, and I think through our studies we started rethinking that and looking at calcium again. So we looked at ways of visualizing calcium, and this work was done in collaboration with Simon Gilroy in the US, and used a probe, GCAMP3, which is based around a GFP sensor, a GFP reporter, and a calmodulin sensor, so that when the calmodulin binds calcium ions, here shown in blue, the calmodulin is absorbed into the GFP sensor, and there is a luminescence. And so this slide here shows you an initial image that we got of an aphid feeding on a plant which was transformed with GCAM3. You'll see a lot of background imaging here from just through expression, but just through overexpression within the plant. But the important thing is when the aphid comes in, that feeding site lights up and shows a calcium signal. And just here's another image, which is maybe even more dramatic. You can see the aphid sitting there on the top of the leaf, and there's the proboscis going through, and down here you can see very beautifully how the calcium signal lights up as the aphid starts its feeding. And I think that's lovely. Well, we needed to get to a more reproducible system than just sitting on an aphid sitting on a plant, so we developed a detached Lethar assay, and you can see here in this image that with the aphid sitting here on the plant leaf, again a lovely calcium image showing in comparison with the control. So there's little doubt then that the aphid feeding will generate a calcium signal. And you can do a lot more experiments then and quantify this, and here's a summary of the signals that we see, and this occurs over a sort of a 5 to 15 minute period with the aphid sitting on the plant and on the plant leaf, showing that the signal is significant. Okay, so where does this signal come from, and what's its significance? So probably not many of you are familiar with insects, as I was not before I began this study, and I just wanted to introduce you to this electrical penetration graph procedure, which helps to tell us how an insect penetrates a plant and where its proboscis is going. And this shows you the basic setup. You fit an electrode to an aphid, you take the electrical signal and send it through an A to D converter and send it down to a computer. And you can then interpret these kinds of this penetration process through the EPG trace in terms of where the aphid is sitting. So here's the EPG trace. It's going, the aphid sitting there, it goes through a pathway phase where it's penetrating the mesophyll, and then it finally reaches the phloem, and at which stage the EPG trace goes rather silent. So we can now use this information to try to understand, going back to our calcium signal, where the signal is coming from as the aphid penetrates the leaf. And what you can see quite clearly here is that the calcium signal is not coming from the phloem. The phloem phase is 20 minutes or beyond the limit of that trace. The signal is coming from the pathway phase, probably from the mesophyll or the epidermal cells. So the critical question then is how is the calcium signal generated? And I just wanted to finish the talk with a few words about calcium channels. So calcium's kept very low in the cytosol. It's much higher at electrochemical potential in the apoplast and in the vacuole. And please don't forget the good old vacuole. And candidate channels that might be responsible for this calcium signal are comprise glutamate receptors, which are located at the plasma membrane, and a channel here which is known as TPC1 or T-pore channel 1, which is located at the vacuole. And so we studied these channels. I don't want to go into a lot of detail here, but we looked at a TPC1 knockout channel, a knockout line, and showed that the signal is the calcium signal is markedly reduced compared with controls. And we got an overactivation line of TPC1, food2, and that shows a much more enhanced calcium signal. So we think that TPC1 and the vacuolar calcium supplies are actually helping to enhance this calcium signal. And we got a similar result with the glutamate receptor mutants. So here you can see in the mutant line very little response in terms of the calcium signal in comparison with controls. And so what that suggests to us is that we've got some calcium coming in from the external medium, the apoplast, and that potentially that calcium is triggering the TPC1 channel to lead to more calcium release. So TPC1 was characterized a number of years ago in the labs of Rainer Hedrich and Julian Schroeder as being a calcium, before the molecular biology was known, as being a calcium activated, calcium permeable channel. And this shows a way in which potentially the calcium signal can be amplified through a little bit of trigger calcium coming in through the plasma membrane, through these glutamate receptors, and then enhancing the calcium release through TPC1, through large calcium stores in the vacuole where it's maintained at millimolar levels. And the theory is that then this intracellular calcium release can go on to elicit calcium plant defense responses against aphid attack. Okay, so just to finish that section of the talk, and then I will have just a few more minutes left. We've shown that aphids elicit calcium elevation in epidermal or mesophyll cells when they begin feeding. The calcium response is initiated by glutamate receptor channels at the plasma membrane, and then we're hypothesizing that the calcium response is amplified through calcium-stimulated calcium release at the vacuolar membrane through this TPC1 channel. Right, in my remaining minutes, I wanted to change the topic somewhat, and that was to talk about plant ecology and essentially why plants grow where they do. And it's about adaptive radiation. And this really focuses on a species of plant called Arabidopsis aeruginosa, not Arabidopsis thaliana. Aeruginosa is somewhat more complicated than thaliana. It's outbreeding rather than inbreeding. It's tetraploid rather than diploid. But it inhabits a wide array of environments, and so there's just a few of these displayed on this slide. So paving stones here, sand dunes by the sea, mountains, and so on. And this work has been a collaboration with my colleague Levi Jans, who's now at Nottingham University, and Frans Martwees, who's at York University. And what I wanted to point out was that where Aeruginosa lives, as I mentioned, it is in a huge variety of habitats. It lives in extremely unfavourable habitats. So here's an example of a serpentine soil, so very high in magnesium and high levels of transition metals, and this is where I came in with my talk, low levels of essential nutrients, N, K and P, and virtually devoid of other plant life. And what Levi Yad did some years ago, and this is the publication at the bottom in PNAS, was he did a GWAS analysis, he and his lab did a GWAS analysis that demonstrated that TPC1, the calcium channel I've been talking about, located at the vacuolar membrane, showed a strong signal which was associated with growth on serpentine soils. We got talking and decided that there may be something interesting there that might be related to the way in which plants can survive in relatively hostile environments. Which is sealed to a that's the magnesium that flow across them. And this has been a finding that's been really exciting to us. You can take that even down to the level of single ionic channels and show that the conductance of those channels is significantly different between vacuoles that are isolated from the serpentine and non-serpentine populations. So this, I think, gives a really intriguing insight into the way in which we can use biophysical and molecular approaches to understand the ecological distributions of plants. So way back 16 years ago, we first characterized the TPC1 channel, and this was the image that we showed in our paper at the time. 12 transmembrane spans and a cytosolic region with EF hands which indicate calcium binding, and that's almost certain to be the region of the calcium-induced calcium release that I talked about earlier. Since that time, things have got a lot more sophisticated, and these TPC1 channels have been crystallized, and we can start to think about how this genomic analysis and the variation in the genomes is reflected in protein structure and function. And this is something that I find really exciting because it moves between biochemistry and right the way through to ecology, and transcends scales essentially. So I just wanted to show before I finish the sequence alignment, and this highlights here that this residue, which is a valine or an isoleucine in all species that have been sequenced with respect to TPC1. So this goes across the range from animals, fish, us, and plants. It's always a valine or an isoleucine, And in all of these serpentine accessions, this is a leucine of all these 946 alleles that have been sequenced. And so the notion that I really wanted to finish on is that we can start to think about using our knowledge of protein structure in terms of addressing how ion channels permeate ions, right the way through from that basic structural question to why plants grow where they do. And I think this is a burgeoning area, and I would encourage more junior colleagues in the audience to think about that as a really exciting prospect that doesn't revolve solely around biochemistry, doesn't revolve solely around the environment, but actually derives from broader thinking. Okay, so just to finish up then, I'm saying that the GWAS analysis of Arabidopsis aeruginosa in terms of serpentine ecotypes shows TPC1 as a major allelic variant. The TPC1 channel in serpentine plants exhibits really different cation transport properties, giving rise to a potential explanation to why these plants are able to grow why they do. And then the differential permeation properties can be used to take into account the wider significance to structure-function relationships of ion channels. Okay, so I'd like to thank you all for your attention, and finishing up, I really need to acknowledge some astonishing collaborators that I've had. The zinc biofortification work was led by Dorina Podar and Paloma Mengue. The aphids and calcium work, I've mentioned Saskia as my colleague who's an entomologist, and Tom Vincent, who was the graduate student who performed the work. And then senior investigators and group leaders Levi Yand and Frans Marthuis, who worked on the TPC1 natural vector, and Tony Miller, a long-standing colleague who has contributed to really all of these projects, and Simon Gilroy from the University of Wisconsin, who collaborated on the GCAM3 work. And funders are acknowledged at the bottom of the slide. So thank you very much, and I would love to receive questions and hope that those can be moderated through the chat function. So thank you everybody for your attention.
Thank you, Del, for a very, very interesting talk, and you took us from your zinc, and you possibly traveled us through the all ecosystem and ended with that in between the aphid story. So it's a huge area that you could cover in only 50 minutes, and there are many more things to learn from you, maybe in future when we could meet physically and we could have many more interaction. And I just was hearing, and I wanted to share with you, if you have seen it and if you know, two of our younger colleagues. One is Jyoti, whom I said, she works on calcium signaling and also on insect. So therefore, we have one lab who is working in insect, which might be interesting to you, and also another very young colleague, and I think is the first alumni of NIPGR who got a faculty position, Hastiram. He just have interest in this zinc and iron and started working in rice. So you have two labs which possibly actually in your area, research area. So thank you, Del, again, and let me see the questions. I saw some of the questions in the chat box. You can also see that, or do you want me to read it out? Whichever way you want me to.
I can see. Thank you so much. Yes, I can see it. Thanks a lot. Many other residues apart from isoleucine and valine are also conserved. Absolutely, and I think this shows the power of genome sequencing across many different ecotypes and different species. And where we go with that, I think, is interesting. But the equally interesting thing is how we interpret this in terms of channel structure and function. So, yeah, sorry, I just saw it. Why focus on, so I have another question. Why focus on zinc? Some other selenium metals have low magnesium, calcium, sorry, I may be misreading this.
Yeah, so there is
Can I pick
one
up
more
with it?
question that you could see, I hope. What happens to the plasma membrane transporters of zinc in the overexpressing lines of MTP?
The short answer is we don't know, but the MTPs are, as Ishod Joshi says, the MTPs are present on the vacuolar membranes. Zinc, we know at the plasma membrane, is largely accumulated by the how those two groups of transporters interact, it really has not been studied, and I think that's an interesting topic. So if I could just say, I'm digressing a little bit and don't want to digress from the other questions, because I think the question's a really interesting one. We have had a project going on in the lab which addresses the whole issue, which I think underpins this question, of how is zinc homeostasis attained? So zinc has to be maintained at very low concentrations in the cytosol. What are the relative concentration? What are the relative contributions of the intracellular and the plasma membrane transporters? We don't know, but we've set up a mutant screen to look at zinc homeostasis defective plants and analyze, try to do a genetic analysis of what are the key components to controlling that homeostasis, essentially through a forward rather than a reverse screen. So we need to address that kind of question through functional analysis.
So next is, why focus on zinc? Some other like selenium metal also more benefit to human and plant.
Yeah. Yes. I'm not saying other elements are not hugely important. I was just struck by the importance of zinc and the number of deaths that zinc deficiency leads to. I didn't mean by focusing on zinc that I think other elements are not hugely important. Selenium is absolutely a hugely important one. as of course is iron. I felt in some senses iron gets a lot of prominence. Maybe certainly selenium doesn't get much prominence in my view, and zinc has had a moderate amount of prominence. So yeah, you can only do one thing at a time, but selenium would be a great area to explore.
So the next one, Athira wants to know, what could insects like aphids feel like feeding on plants growing in serpentine soil and which have low magnesium and calcium ratio?
Yeah, that's a great question. I hadn't thought of testing that one. Yeah, we should do that experiment. That would be a good thing to do. And as I said, Arabidopsis aeruginosa it's rather different from Thaliana. I don't honestly know whether the aphid species we're working on will feed on Arenosa, but my guess is since the species we were are working with is a promiscuous feeder, it would feed on Arenosa, and that would be great to look at. And what a stimulating question. Yeah.
So I just would like, so is there anyone from audience who are here wants to have any question? Okay, so just a moment, Del. I'll tell you if someone is interested. So let's see. You are not.
Del, it's a fantastic talk. I have a question. So under stressful conditions, for example, if you give heavy metal stress to the plants, and there is a increase in levels of the reactive oxygen species and also reactive nitrogen species, do you think any of these species can actually modify these transporters, like PTMs, any kind of PTMs? Those play important role in determining activity of these transporters, in turn actually determine the transport. So are there any evidence on that?
Could you hear the question, Del, or should I repeat?
Super, I didn't hear any of that. I'm so sorry.
Okay,
Could you
so
repeat
the question is that there are a lot of reactive oxygen species and reactive nitrogen species. So is there any evidence that whether this kind of species can also have some kind of effect on those transporters onto their PTMs, and that might be related to the, transporting the ions?
Yes, I think there is evidence of that, and as the questioner said, there is a lot of evidence that reactive oxygen species are involved in defense signaling from aphids, and that includes a role of jasmonate too. I think there are isolated reports of ROS acting on membrane transporters, but again, I think that integrated approach has not really been has not really been elucidated, and I think it's really another rich area where for research. But great question.
Yeah. Any other questions, Jyoti? Okay, so my last, it's not a kind of question, but I was just inquisitive to understand that from the zinc enriched cereals, I don't know whether you have undertaken any kind of work in a model of finding out if you have a more zinc, because we know that human also has zinc transporters, right, in the liver and the intestine. So have you tried to look into it? That and we know those transporters in the human cells are also regulated by the level of the zinc that you have. So is there anything that you have looked into it? And if so, then whether the intestinal zinc transporters or the liver zinc transporter would have more impact on the bioavailability of the zinc in the human body?
I think that the bioavailability question limits its availability, and I think an integrated approach again to developing enhancing zinc nutrition involves not only understanding zinc but how it's complexed and made bioavailable for human transporters in the gut. So that's ongoing work.
Okay, so I think there is no more question, and I really thank you, Del, for sparing your very valuable time and to be with us for almost 1.5 hours. So after this, basically we would have some kind of a prize distribution, because this is the day when we actually give away the prizes. Had you been here, you would have done this, but I don't know how much time is left for you. You can still be with us virtually, and we would now start that for our students of the second year and the fourth year PhD students get some prizes. We give these seminars, and the best of the talk comes, and other one is kind of a student who publishes the best paper of the year, and also there are some other prizes which would be announced now, and I hope you can stay with us, and that will be really inspiring to the students. So with that, I now hand over, and maybe Mr. Datta will take it forward. So thank you, Del, and we will be in touch. And
thank you very much.
Yeah, look
thank you very
forward
much,
to
Subra. I
welcome
will
you.
certainly stay. Yes, and it's been a tremendous honour, I will say again, to be able to talk today about science with you. And
Yeah.
I very much hope to visit you soon.
Thank you. As if that he is giving, so I'll just give away the prizes. That's the way.
Okay, very good afternoon to everyone. As always, I take this esteemed privilege to announce some of the awards and rewards for our students who have excelled in the field of academics and co-curricular activities. On this special day, we would like to appreciate and encourage our students who have worked hard and performed well, especially during their coursework examination. As you may be aware, that Institute has been conducting its PhD program in affiliation. So as you may be aware, that Institute has been conducting its PhD program in affiliation with JNU since 2001. Just for statistics, till date, around 350 students have been enrolled in this program, with annual intake of about 25 students. Currently, 146 students are continuing their PhD. Till date, NIPGR has produced 158 PhDs. In the first year of their PhD program, the students undergo a coursework and the end of which they have academically performance is evaluated. Today, we would like to present a medallion and a certificate of merit to the student who performed best in the academic session 2020-21. Ladies and gentlemen, please put your hand together for Prakashi Anecha. Now, Prakashi has scored a cumulative grade point average of 9 on a 10-point scale from 0 to 9. Another event that was selected a couple of years back and has become very popular is the SciFlex. The symposium consists of presentations by the second-year students on their project theme, followed by presentations by the fourth-year students who discuss the progress of their research work. The event provided a unique opportunity for the students to showcase their talent, since this two-day event is managed by the representatives chosen among the students as convener and co-convener. Usually it ends with a cultural program Rainbow prepared by the students, but this year, due to this pandemic, culture program could not happen. Now, coming back to the presentations, a panel of scientists evaluate all the presentations, and the best scientific presentations are rewarded. The medal and certificate for the best scientific presentation at SciFlex 2021 in the respective groups goes to Shruti Mishra for the second year, and Ashish Prasad for the fourth year. Now, in this series, another event that we have started recently is to reward the best scientific research papers for the academic year. The research article published by the research group of the Institute are evaluated by the committee, and best publication is selected. The first author of the publication also presents the research work in the seminar organized by the Institute. For the academic session 2021, 3 research papers are selected for this category, and the honors goes to Sunil Kumar Yadav for the first paper, Jyoti May Mathan for the second paper, Deepti Nambiar and Juhi Kumari for the third paper. Now, we would also like to facilitate the conveners of SciFlex to appreciate their effort who made this event a success. Ladies and gentlemen, please welcome Vishal Vashane and Rekha Jaiswal. Okay, Vishal and Rekha, please come on stage and take your presence. All right, moving on to the next stream is our Hindi Pakhwara. Hindi was accepted as official language of our Union of India on 14 September 1949. To commemorate this occasion, Hindi Divas and Hindi Pakhwara is celebrated every year in this institute. Among other events, this year on the occasion of Hindi Divas, we have organized a Hindi Nibandh Pratyogita and Hindi Kavya Path. Staff members, students and researchers have participated enthusiastically in these events. A committee has evaluated and selected 2 best essays and 2 winners of the Kavya Path event. For Hindi Nibandh Pratyogita, the honors goes to Samar Singh for the first prize, and Dr. Vinod Kumar Sharma for the second prize. And the winners for the Hindi Kavya part is Pushpendra Kumar Mishra for the first prize. And Prakashi Aneja for the second prize. Well, these were the winners, and the participation certificate will be given to all the participants. All right. Actually, okay. So now Institute is maintaining a very high standards of implementation of official language policy of the Government of India for promoting progressive use of Hindi in the Institute. In this series, Institute is publishing its Hindi magazine, we call it Anvekshan, which consists of research abstracts, essays, poetry, stories, jokes, etc. in Hindi, contributed by staff members, researchers, and students of the Institute. Okay. So we will be, the third edition of the Hindi magazine is compiled, and today we are going to release the third edition. If the members of the Hindi committee are present, I request them please come on the stage and join Director Madam for releasing this Hindi magazine. Yes. So Dr. Amarpal Singh, Dr. Pinky Agarwal and Dr. Gopalji Jha are our members in the Hindi committee. And with their sincere efforts, this booklet has come up. So many thanks for releasing this booklet. Now I request to take your seats. So now we are moving to the next event. So now I invite Dr. Alok Sinha, our senior scientist of the Institute, to propose a formal vote of thanks. Thank you.
Yeah. A very good evening to all present in this hall and to all those who are connected with us online from different parts of the country. And a very good morning to our special guest and speaker of the day from Norway. So it is indeed a privilege to propose this vote of thanks on the completion of this 24th Foundation Day program of our loving institute NIPGR. I, on behalf of entire NIPGR family and on my personal behalf, express our gratitude and thanks to the speaker, our distinguished speaker of today's, Professor Del Sanders, for gracing this occasion and sharing with his findings and opinions today. Believe me, Professor Sanders, that your talk has motivated one and all in this audience and all, I'm sure, connected with us through online. We loved hearing your zinc transporter, then you have this fancy EPG recording for calcium, and then why do plants grow where they do. So it's a very, very interesting, and it has motivated one and all, and I'm sure many of our younger colleagues are already planning to start a program in these lines. Thank you very much for that. I once again thank you, Professor Sanders, for furthering our celebration with your visionary perspective and sparing your valuable time from a very, very busy schedule. I would like to thank and express sincere thanks to our director, Dr. Subra Chakravarti, for her consistent encouragement, guidance, and positive outlook. We appreciate, Madam, your consistent efforts to attract more funding for research, and we look forward to your leadership to help us to serve and give us our best to the science. I thank my friend and colleague, Dr. Sabita Bhatia, for conducting today's function so smoothly and seamlessly. I also extend my thanks to my fellow colleague scientists and entire NIPGR administration led by Mr. Sandeep Datta for their enormous cooperation in the organization of this event, and their efforts are really appreciated. I would like to take this opportunity to express our deep sense of gratitude and appreciation for all our distinguished guests who joined here in our celebration in this hall, guests from DBT, neighboring institutes and universities, and also those who are connected with us through online from different parts of the country. Last but not least, I would like to thank our NIPGR students and researchers for their active and supporting role in organizing this event. I congratulate all the students for the excellent performance throughout the year, including those who have won awards today. It is you, dear students, who make NIPGR what it is today. So I thank you all once again. With this, we come to the end of this program today, and I invite all who are present in this hall and those who are present in the institute to come out for a cup of tea outside. We are going to miss you, Del, for this tea. Maybe sometimes in future, we may have you here in physical and we can interact more with you. Thank you very much for your time. Yeah, thank you. So we can log off now? Yeah, we can log off and end the program now. Thank you very much.